our next speaker is Dr. Marie Davis.
Thank you.
And so we've heard quite a bit about GBA already, and Dr. Davis is a basic scientist as well as a neurologist in movement disorders, and who's going to dive deeper into the basic science of GBA. So, Dr. Davis.
Hi, can everyone hear me? So, as Dr. Chuang said, I'm a basic scientist and also a movement disorder neurologist. So I see movement disorder patients at the Seattle VA, and I also see movement disorder patients that have a genetic basis or familial inherited type of movement disorder at the University of Washington in the neurogenetics clinic. So I'm going to tell you about the work that I'm doing in the lab right now. In my lab, I'm currently moving it to the, I'm starting my own lab at the Seattle VA. But I've been doing work at University of Washington so far. So you've already heard a lot about genetics in Parkinson's, and I'll just briefly say, remind that there's a lot of different genetic influences in Parkinson's, and we now know that it's more of a spectrum and a lot more complicated than we previously thought. So there are, let's see. There we go. There are what we call single gene Mendelian causes. If you have a mutation in one of these genes, you're expected pretty much to develop Parkinson's during your lifetime. But these are really rare. They make up less than 10% of all Parkinson's cases. So they're very, very, very rare. But they can give us some insight into the disease. But you know, we're not sure how applicable they are to all Parkinson's cases. More recently, we found also in more of the general population much more common genetic influences through large studies of thousands and thousands of patients. Genetics in Parkinson's is much more complex, and we have very strong genetic factors as well as really weak ones that are much more common in all people, but the effect in increasing your risk of Parkinson's with these more common factors is very, very small. So at this point, it's not very clinically relevant. We don't test people clinically and say, oh, you have this factor, because they increase your risk by such a small amount. So these are the really strong factors, but they're very rare, so they're not in most Parkinson's patients. But we now have a very strong risk factor, and you've heard a lot about it so far today, and that's GBA. And so I've been focusing my work trying to understand how GBA increases the risk of Parkinson's and how that might give us some insight on how Parkinson's is caused in everybody, not just the people who have a mutation in GBA. So you've already heard a lot about GBA, but I'll just say one more time. When you have a mutation in GBA, it increases your risk of Parkinson's compared to people who don't have a mutation in GBA. When you have a mutation in GBA, it also increases your risk of cognitive impairment, and also you have a faster progression of Parkinson's disease. So it's a really important gene in how Parkinson's develops and how it progresses, and we're trying to understand what it is about GBA that can influence Parkinson's. So GBA is an enzyme, and so it breaks down a lipid that has a sugar attached to it. So this is what GBA breaks down, and it breaks it into the sugar by itself as well as the rest of the lipid, which is a really important lipid in terms of the health of cells, and especially neurons. So the way that I'm looking into how GBA influences Parkinson's disease and leads to this disease is through two systems. So I'm using fruit flies and also human neurons. And whoops, sorry. So you might wonder why I would choose fruit flies. So we're very different from fruit flies, but we actually have a bunch of similarities. So a lot of the genes in fruit flies are also, we have equivalent genes in us, and about 75% of the genes in humans that cause disease have an equivalent in fruit flies. So we do have a lot of similarity. We have a lot of genetic tools in fruit flies that we can use that are readily available. And fruit flies, unlike cells, are a whole organism. We can test them for different deficits, including movement deficits. So I do put them through a climbing assay and see if they have a movement disorder. I can even do an assay for their cognition that I'm happy to talk about more. And then fruit flies live a pretty short life. So in the lab, at most, they live about 3 to 4 months. And so when we're looking at studying diseases that are mainly occurring later in life, it's nice to have a short-lived organism rather than something like a mouse where you have to wait, maybe a year or several months. So there's a lot of good reasons to use flies, but they're not perfect. And so that's why I'm also complementing it with a cell culture system that's based on neurons derived from humans. So the way that I do this is I'm taking advantage of a newer technology where we can get cells from your skin or some other readily available tissue, and then reprogram those cells to become more primitive, as if it was like in an early embryo, and then differentiate them back into the tissue of interest. So for me, that's really neurons and sometimes the support cells of neurons. And so I can basically make neurons from a patient, and that's a really new, powerful tool that a lot of people are now using, because we don't really do brain biopsy, or we try not to do brain biopsies and actually get neuronal tissue. From most of the time, that's not really an option. So it's a useful tool for us. So this human neuronal model is nice because it's a little bit more relevant to us compared to the fruit flies. And we can look at neuron-specific effects because although flies do have neurons, they're very small. They're a lot harder to study individually. So I just want to tell you briefly the things that I'm looking at right now. I'm going to skip over this. So as Dr. Zabedian and Dr. Aldred mentioned, talked about quite a bit, we think that Parkinson's can be due to the accumulation of proteins that are clumping in neurons, and it's leading to dysfunction of those neurons. And we definitely see that in when there's loss of function of GBA. But I've been studying some new results suggesting that these protein clumps.
If anybody, are you looking for any volunteers to give iPS cells for skin biopsies?
So yes, I am actually, and I collaborate with Cyrus Zabedian. So he has a repository of tissue through his Parkinson's genetic research study, and actually the neurons that I'm using now derived from a patient that was through the PAGER study. So yeah, the PAGER study is a really good source of for this research.
Are there specific symptoms that you're looking to target in your research?
So at this point, it's not really a symptom based research, but what I'm trying to do is understand the actual root cause of Parkinson's through these sort of genetic inroads. And with these genetic models in fruit flies and cells, we hope that we can eventually translate these findings to to patients, and also potentially test drugs at a preclinical stage in these models.
So it kind of goes back to the idea of, someone asked earlier about, how does one person differ from another person in terms of progression? We've already talked about the GBA, and Dr. Davis is looking at the root cause and why does someone with Parkinson's develop dementia? And so in one of her slides, she kind of pointed out the interaction between perhaps GBA and Lewy body. And so that's important as well. So that's the cutting edge of the basic science there. So thanks, Dr. Davis.